D-Dive 345. 1971, Dr. Ruth Turner descended in Alvin, becoming the first woman scientist to observe the ocean floor through a submersible's viewport. The marine biologist specialized in wood-boring mollusks, organisms that seemed impossibly specialized until Turner demonstrated their critical role in deep-sea ecosystems. Her presence in that titanium sphere represented more than personal achievement. It shattered assumptions about who belonged in extreme environments, who could conduct cutting-edge research, who deserved access to frontier exploration. Turner's scientific contributions would prove immense. Her woodfall research revealed how organic debris creates chemosynthetic habitat corridors. But that first dive's significance transcended any single discovery. It established precedent. Capability determines access, not gender. Every woman scientist who has descended since, every barrier broken in ocean research, traces back to Turner, proving that excellence, not demographics, should govern who explores the deep. Ruth Turner built her career studying organisms most scientists dismissed as curiosities, wood-boring bivalves, while colleagues pursued charismatic megafauna or commercially important species. Turner focused on Xylophaga and related mollusks that colonized sunken wood in deep-sea environments. Her choice seemed perversely specialized, deliberately selecting the obscure over the obvious, the marginal over the mainstream. But Turner understood something others missed. Specialized organisms often played outsized ecological roles, precisely because their specialization filled niches nothing else exploited. Wood-boring bivalves didn't merely consume dead wood, they transformed it, creating sulfidic microenvironments colonizable by chemosynthetic bacteria. The bacteria supported communities resembling those at hydrothermal vents, minus the volcanic heat source. Turner hypothesized that wood falls, whale falls, and other organic debris acted as stepping stones enabling chemosynthetic species dispersal between isolated vent sites. The hypothesis required direct observation of deep-sea wood colonization. Surface ship operations could deploy wood samples on seafloor, recover them months later, analyze what colonized them. But that approach missed process details. How quickly did bivalves locate and colonize wood? Did colonization patterns vary with depth, temperature, or proximity to other chemosynthetic habitats? What succession occurred? Did pioneering species prepare wood for later colonizers? Modern imaging technology would eventually address some questions through autonomous systems. Cameras merging multiple exposures into photomosaics mapped habitats from centimeters to hundreds of meters resolution. Tocam deployments using 24-megapixel Nikon D3 300 digital single-lens reflex cameras with dome optics rated to 6,000 meters created detailed seafloor maps. Miso Oi's GoPro cameras provided 12-megapixel still capability with customizable intervals, enabling time-lapse documentation of colonization sequences. IP multi-seacom systems on 5 deepslanders reached 11,000 meters, proving that imaging technology could document life at any ocean depth. But in 1971, before digital imaging revolution, before autonomous camera systems, before high-resolution photomosaics, direct human observation remained paramount. Turner needed to see woodfall sites personally, make real-time decisions about sampling priorities, recognize unexpected patterns requiring investigation. Remote sensing couldn't replicate trained, expert identifying significant observations amid overwhelming sensory information. Turner's expertise positioned her perfectly for submersible research. She knew what mattered. She recognized patterns indicating ecological processes. She asked questions others wouldn't formulate. Yet institutional barriers prevented her access to tools her research required. Not because she lacked qualifications, not because her science was questionable, but because deep-sea exploration remained male-dominated field where informal rules excluded women regardless of merit. The barriers weren't explicit policies. No written regulations forbade women from submersible operations. Instead, Unstated assumptions created an environment where women's participation seemed improbable, inappropriate, or practically impossible. Equipment designed for male bodies, logistical arrangements assuming male crew, cultural norms treating women scientists as exceptions requiring special accommodation rather than colleagues deserving equal access. These informal barriers proved more insidious than formal rules because they operated through countless small decisions rather than single policy addressable through formal challenge. Turner navigated these barriers through scientific excellence so obvious that excluding her became indefensible. Her wood-boring bivalve expertise was unique. Her research questions were compelling. Her proposed observations would advance understanding. Excluding her meant denying science valuable data because investigator happened to be female. Eventually, institutional resistance yielded to scientific merit. D-Dive 345, 1971, marked more than calendar date or sequential dive number. It represented institutional acknowledgement that capability, not gender, should determine access to research tools. Dr. Ruth Turner became first female scientific observer aboard Alvin, not through quota or special program, but through recognition that her expertise warranted submersible access. The significance lay precisely in how matter-of-fact the dive should have been. Turner possessed relevant expertise. Her research required direct observation. Submersibles enabled that observation. Therefore, Turner should descend. Simple logic, yet revolutionary in field where women's participation remained exceptional rather than normal. Turner approached the dive as work, not historic milestone. She had research questions needing answers. Submersibles provided tools answering those questions. Personal significance mattered less than scientific objectives. This pragmatic focus itself challenged assumptions. Women weren't fragile creatures requiring special consideration, but colleagues treating extreme environments as routine research venues. Modern autonomous imaging would later reduce need for human presence in some research applications. Quiescent imaging freed pilots from camera operation duties through pre-programmed photography. 2016 Oasis expedition used 24-megapixel cameras at 10-second intervals, generating comprehensive habitat documentation without human intervention. 2018 Loki Castle photo mosaic combined 263 images mapping approximately 100-meter scarp feature. 2019 5 Deeps expedition biodiversity documentation led to new species identification within weeks. Remarkable speed demonstrating advancing technology's capability. But Turner's dive preceded this technological revolution. In 1971, human observers remained irreplaceable. She watched wood-boring bivalves colonizing experimental substrates, noted patterns in distribution and abundance, collected specimens requiring later laboratory analysis. Her observations validated theories about woodfall ecosystems that remote sampling alone couldn't confirm. Context mattered. Real-time judgment mattered. Expert interpretation mattered. The dive established precedent extending beyond Turner personally. If one woman scientist merited submersible access based on expertise, logic demanded all qualified women scientists receive similar access. The barrier broke not through dramatic confrontation, but through demonstration that excluding women diminished science itself. Turner's contributions proved valuable. Her observations advanced understanding. Her presence aboard Alvin caused no problems predicted by those who'd resisted women's participation. The barrier-breaking dive's success made subsequent barriers harder to justify. Institutional change rarely occurs through single dramatic event. More often, precedents accumulate until old assumptions become untenable. Turner's dive contributed to accumulation, establishing that deep-sea exploration welcomes all qualified researchers regardless of gender. Each subsequent woman scientist descended more easily because Turner proved participation worked. Turner's research revealed that sunken wood created surprisingly complex ecosystems in environments seemingly barren of life. Deep seafloor away from hydrothermal vents typically supports sparse populations adapted to scarce food resources. Organic debris falling from surface, dead organisms, fecal pellets, plant material, provides most nutrition. Wood represents particularly concentrated organic resource. But most deep-sea organisms can't digest cellulose or linen comprising wood structure. Wood-boring bivalves evolved specialized capabilities exploiting this resource. They bored into wood mechanically, harbored symbiotic bacteria digesting cellulose, created tunnels and galleries transforming solid wood into porous substrate. As wood decayed, sulfate-reducing bacteria colonized oxygen-depleted microenvironments, producing hydrogen sulfide. Chemosynthetic bacteria oxidizing hydrogen sulfide established, supporting communities including clams, mussels, and tube worms resembling vent fauna. The Mood Fall revelation changed understanding of how chemosynthetic species maintained genetic connectivity between isolated vent sites. Vents occurred sporadically along mid-ocean ridges, separated by tens to hundreds of kilometers. Individual vents operated for decades or centuries before geological changes shut them down. New vents opened elsewhere. Vent-dependent species needed migration pathways reaching new sites before old sites became uninhabitable. Turner's research suggested wood falls, whale falls, and other organic debris created temporary chemosynthetic habitats serving as stepping stones. Larvae dispersing from established vents could colonize wood falls, mature, reproduce, and send their larvae to next stepping stone. The network of temporary habitats enabled genetic exchange across distances species couldn't traverse directly. Modern understanding recognizes multiple chemosynthetic habitat types, chemosynthesis. Theoretically proposed 1890 by Sergei Nikolaevich Vinogradsky, describes metabolism, where microbes extract energy from inorganic chemical reactions rather than photosynthesis, or consuming photosynthetically derived organic matter. Chemosynthetic microbes oxidize reduced compounds like methane or hydrogen sulfide, capturing released energy to fix carbon dioxide into organic molecules. These primary producers support entire ecosystems independent from sunlight. Different chemosynthetic environments offer varying durations. Hydrothermal vents operate decades to centuries. Cold seeps may persist millennia. Whale falls provide resources for years to decades depending on carcass size. Wood falls last months to years. The temporal mosaic creates complex dispersal dynamics where species adapted to ephemeral habitats maintain connectivity through colonization and reproduction cycles timed to stepping stone availability. Turner's specialized focus on seemingly obscure organisms revealed ecosystem connectivity patterns applicable far beyond wood-boring bivalves. The principle that specialized species often play disproportionate ecological roles extended to understanding how biodiversity maintains itself, how ecosystems respond to disturbance, how conservation should prioritize protecting not just charismatic species, but also specialists enabling community function. The bopper between Turner's scientific approach and her barrier-breaking role emerged clearly. Both cases involved recognizing that dismissing specialists as marginal missed their critical contributions. Wood-boring bivalves weren't evolutionary curiosities, but ecosystem engineers. Women scientists weren't exceptional cases requiring special accommodation, but colleagues whose perspectives and expertise enriched research. In both instances, expanding what was considered valuable improved outcomes. Turner's dive opened doors remaining open since. Women scientists descended in Alvin routinely, their participation unremarkable precisely as it should be. The normalization represented ultimate success, not celebrated exceptionalism, but matter-of-fact inclusion based on scientific merit. Modern ocean science exhibits gender balance reflecting Turner's legacy combined with broader societal changes. Research cruises, submersible operations, laboratory analyses, all include women scientists as colleagues rather than exceptions. The transformation took decades, required persistent effort from many individuals, benefited from institutional policy changes. But Turner's 1971 dive established early precedent proving that capability determined who should explore the deep. Her scientific legacy continued expanding beyond single barrier-breaking dive. Woodfall research revealed ecosystem connectivity, enabling species persistence. Her methods influenced how marine biologists studied colonization processes, succession dynamics, habitat utilization. The specialized focus, initially seeming narrow, proved broadly applicable. Understanding specific systems deeply often reveals general principles operating across ecosystems. The dual legacy: scientific discovery and institutional change. Intertwined rather than separated. Turner's research excellence justified her submersible access. Her submersible access enabled research discoveries. Her discoveries validated expertise meriting access. The cycle demonstrated that including previously excluded voices improved science through adding perspectives and expertise missing from homogeneous research communities. Contemporary women oceanographers working globally trace professional lineage partly to Turner. Not always directly. Many never met her. Studied different topics. Worked different regions. But precedent she established created an environment where women's participation in ocean research became normal rather than exceptional. The quiet revolution she advanced didn't demand recognition but demanded access. Insisted that excellence be sole criterion determining who explored frontiers. Demonstrated through achievement that expanding participation improved science. To Turner's barrier breaking remains relevant because frontiers persist where access remains unequal. Whether in deep ocean exploration, space missions, Antarctic research, or any domain requiring significant resources and institutional support. Ensuring that capability rather than demographics determines participation requires ongoing attention. Turner's example reminds that progress occurs through demonstrating that excluded groups contribute meaningfully when given opportunity. The A.I. Wood boring bivalve specialist who descended in 1971 proved that organisms dismissed as marginal often play critical roles. That scientists dismissed as exceptions often provide essential perspectives. That excellent work speaks for itself when institutions allow it voice. Her legacy persists in every woman scientist conducting deep ocean research, in every understanding of how chemosynthetic ecosystems maintain connectivity, in every recognition that expanding who participates in exploration expands what exploration discovers.